Hi guys, welcome to the Unit 4 review video. It's very simple. The questions on this PowerPoint will also match the questions on the handout. I'm going to explain the answers and tell you what they are. I'm going to count down for 5 seconds, showing just the question. After 5 seconds, I show you the answer and explain it. Of course, if you need more time, which you may, pause the video as needed. And then if something is wrong on your handout, you need to not only change your answer, but write down some key things I say to help you remember it. All right, let's go ahead and begin. In an experiment, Selmer puts a high concentration of solutes on side B of the bucket and a few solutes on side A. If water is added to the bucket, where would water first move across the semi-permeable membrane? You have 5 seconds. Okay, the answer to this is A. Water will flow from side A to side B. And the reason is water moves to the highest solute. So I look and say, well, where's the most solute? It's on side B. There's a whole bunch here, a few here. So what is water going to do? It's going to move on the side where there's more solute in that side B, which makes the answer A. All right, next question. Once the solution across a membrane reaches dynamic equilibrium, the molecules in the solution will what? You have 5 seconds. All right, the answer to this is A. It will continue to move across the membrane in both directions. If I look right here, we can see that it's at equilibrium. There's 3 solutes inside the cell, 3 solutes in the solution. So here's the thing, it never stops. It's going to keep moving, but as one leaves, one enters. So it still makes it balanced, but it never stops moving. So never choose C, because equilibrium, there's always moving molecules. It just always stays equal. All right, question 3. Why does misting vegetables help them look fresh? 5 seconds. All right, the answer is the vacuoles fill with water by osmosis. So think about it like this. We spray plants so they can get enough water, and they hold it in their vacuoles, which helps the plant look more fresh and more alive. Well, our vegetables and all that kind of stuff are forms of plants. So the grocers spray it with water so that it can look more fresh so that people will buy it. But the only reason they look fresh is because the vacuoles fill with water. All right, next question. Identify the diagram as either hypotonic, hypertonic, or isotonic. You have 5 seconds. All right, so the first diagram is hypertonic. I know because it's shriveled up. Why does it shrivel? There is more solute in the solution, which is outside the cell. There's more solute there. So where does water move? To the highest solute, which means water is going to leave the cell and go to the solution or the outside because there's more solute there. All right, the second one is isotonic. And this is where it's equal. So the cell stays the same, nothing changes about it. And there's more solute. Sorry, the solute is equal in both the cell and the solution, which means the cell stays the same, right? As one enters, one leaves, and that is equilibrium. Okay, and then the third one is hypotonic. So we notice the cells are a lot more bigger than the original one. And that means that water is entering the cell. Well, why does water enter? There's a high concentration of solute inside of that cell, which means the water is going to flow in, and there's a possibility it might rupture as well. All right, next question. So match the function of the cell membrane components to their correct description. 5 seconds. All right, the answer is D, C, A, B. I know it's carbohydrates because it says that it's identifying chemical signals outside the cell. Think about the carbohydrates like an antenna. It's always sending out signals and trying to receive signals so that it can help the cell or help the TV, right, get the connection it needs. Carbohydrates help the cell get what it needs. Then we have cholesterol. We know cholesterol is nonpolar because it's located between the phospholipids and the bilayer, and that middle portion is nonpolar. So nonpolar is with nonpolar. And cholesterol helps maintain fluidity and stability. Remember that as well. Phospholipid bilayer has the polar and nonpolar ends, and they help to form the bilayer of the plasma membrane and transport proteins, of course. You have two types. You have channel proteins and carrier helps pump it, and they help to move molecules across the membrane that can't pass on their own. All right, next one. Identify the structures in the cell membrane below. 5 seconds. All right, and the answer is carbohydrates for A, B is phospholipid bilayer, C is carbohydrates. Again, it looks like an antenna just like it would be on a TV. They help with signaling, so A is your carbohydrates, B is the bilayer, basically makes up the largest component of the cell membrane, and C is protein. Yes, I know it's kind of crazy because they put it sideways, but if you look here, this is the same picture as that, and it's a transport protein. Now, give me the 3 names for this diagram. 5 seconds. All right. Cell membrane, plasma membrane, fluid mosaic model. Those are the 3 names for this picture right here. All right, next question. In an experiment, a cell was placed in an isotonic solution. Then additional solutes was added to the solution. What will happen to the cell? 5 seconds. One. Okay. All right, so I wanted to label this real quick, or annotate it. And basically, of course, we know it's isotonic. That means the solute is equal in the cell and out of the cell. But here's the thing. Someone added solutes where? To the solution. So just labeling our diagram. Oops. This right here is our cell. And this right here is our solution. And we know that they added solutes to the solution. So I'm going to say added sol. I'm going to put a U, solutes, right? Here. So it's higher here. And I'm going to box that because this solute is higher. And if this is the highest solute, where does water move? To the highest solute, which means water is going to leave the cell and go to the solution because it has a higher amount of solute, which means if water leaves, the cell shrinks, just like if you release air from a balloon. So the answer to this would actually be B, that the cell will shrink, which actually makes it a hypertonic solution. Good job if you get it right. All right, next question. The following statement was made by a student. Which mode of transport are they referring to? I noticed the plasma membrane was pumping excess ions out of the cell into an environment where there is a higher concentration of ions. You have 5 seconds. All right. So as soon as I find the little mouse pin, there we go. So it says it is pumping excess ions into a higher concentration. So if it's going in a higher concentration, it's going to a higher one, that means it's coming from a lower concentration. And if it's going from low to high, that lets me know it's active transport, and I'm going to put AT for active transport. Another indicator, and a huge one, is the pumping. So if it's pumping, that means it's using ATP. So if it's active transport and it's using ATP, I only have one option: protein pump. Okay? I know it's not diffusion, facilitated, or osmosis because those are passive and they do not use ATP. And I know it's protein pump because it is pumping and it is protein pump. Awesome. Okay. Which of these modes of transport uses protein? Again, which of these modes of transport uses protein? 5 seconds. Okay, the answer to this is B and E. So, facilitated diffusion uses proteins, and of course, it is passive transport. And protein pump uses proteins, but it's active. So basically, each type of transport uses protein in one way or another. Okay, now I want you to match the description of these words to their descriptions. 5 seconds. Awesome. So the answer to this is B, A, E, C, F, D. So I'm looking here at this one. It says that transport proteins are moving molecules across the membrane. Well, first, it doesn't tell me that it's using energy. And whenever they want us to know it's active, they tell us it's using energy. So I know automatically that this is passive transport. And if it's passive transport, I only have so many options because only one passive transport uses proteins, and that's facilitated diffusion, which is B. The second one, molecules can readily pass, meaning without any help. They can just pass through the membrane without the use or need of ATP, which makes my answer A, diffusion. Diffusion, it passes through. It doesn't need proteins, it doesn't need energy, and of course, since I know it doesn't need energy, I know off the rip that it is passive transport. Now, if it was osmosis, they would have told me it was water. But since they didn't, I know it was diffusion. All right, the third one. Molecules are pumped from a lower to a higher concentration. And the word pumped again. So whenever I say that word pumped, I'm automatically knowing that it is using ATP, which makes this definitely an active transport. And I know that anyways because it says it's moving from low to high or against the gradient, which is active. And if it's pumping it, it's protein pump E. All right, the next one, it engulfs. Engulfs. It sounds a lot like it's going in. So if it's going into the cell and it's a bulk of material, that makes it endocytosis. The next one is that it releases a bulk of material. If it releases it, it's exo. And of course, if it's moving water without ATP, that makes it osmosis. Next question. Which statement best explains why the cell membrane is described as semi-permeable? 5 seconds. Awesome. The answer is A. Semi-permeable means it controls what enters and exits the cell. So basically, it's like a bouncer at a club. I'm only letting certain people in, and I'm letting some people get out. All right, next question. Which statement about cholesterol is correct? 5 seconds. All right, the answer is statement 4. Cholesterol is nonpolar because it is located between a phospholipid bilayer, and this is true all the way around. Next question. What will happen to the cell in an isotonic solution, hypotonic solution, and hypertonic solution? 5 seconds. All right. In isotonic, iso means equal, so the cell will stay the same. And hypotonic is hippo, hypo. So the cell is going to swell, and if it swells too much, it will rupture. In hypertonic, the cell is going to shrivel or shrink. Next question. The osmotic solution is displayed to the right, the bottom. How could you label this figure and explain how you knew? All right, 5 seconds. All right, the answer is D, plasmolysis, and you know because I told you, but also because we see the cell inside is wilting or shrinking, so it's plasmolysis, and it's a plant cell because it's square. All right, last one. Is a diagram endo or exocytosis, and explain how you knew? 5 seconds. Awesome. So it is endocytosis, and I know the first one because it is showing the molecules going in, the arrows are pointing, and then in this one, they're showing them pointing out. So that is it. I wanted to make sure the video was under 15 minutes.